So our main speaker this evening is Professor Stephen Baker, Director of Research based in the Department of Medicine at Cambridge, and Honorary Professor at the University of Oxford. And you're going to tell us about where we go next with antimicrobial resistance. I hope this is a good news story. I mean, it's
let's see.
With governments falling in and out of fashion, it's nice to have some real world stuff, I'm sure. Right, okay.
Thank you very much. Thanks for inviting me to talk this evening. What an honor to be here. And I'm glad some people have made it out into the cold autumn night to come and listen. Thank you for turning up. So this is going to be a fairly rambling talk, ranging from some kind of popular science bits to some of the stuff that we're also doing in a laboratory. And I make no apologies for that. The way we kind of construct our science is kind of slightly different to what you've heard before, with the way we focus on many different things to try and tackle bigger problems. So I'm based here at the JCBC, we call it, on the biomedical campus in Cambridge, which is a slightly different environment to the one that I'm used to. So part of the reason really I'm interested in kind of global health research is I spent 12 years living and working in Asia, in Ho Chi Minh City. And this was a real turning point in my kind of scientific career of understanding not just really about how kind of real infectious disease research goes, but actually being involved in it in real time. So drug-resistant bacteria is something we've been faced with really since the dawn of antibiotics, but it's really in the last 15 years, it's gone through a rapid acceleration in the way not only they've emerged and spread and become dominant, but also in the techniques we use to understand them. So being in the opportunity to work somewhere like that and use some of these techniques in a real life location was very rewarding and very exciting. So I returned back from Ho Chi Minh City in 2019 with a view of setting up a research group. But I'm not sure whether you've seen the news, but there was a pandemic around the beginning of 2020, which was also of interesting to me as a kind of infectious disease researcher. I'm not a virologist, but having the experience of working in a research hospital, in an infectious disease hospital, we were able to pivot the group relatively quickly. And you may have seen this at the time, that we were able to then turn the laboratory in a space of 5 days into the first academic diagnostic facility in the country. So this was a real achievement and a massive, massive effort at the time. We were working 12 hours a day up until the summer of 2020. There's a picture of Ian there swabbing, which I won't show you. But also then a kind of interesting academic inflection as well, of understanding not only how you can do things that are useful during a pandemic, but also it became a kind of revelation for understanding how you could do applied research in such an environment in such a short space of time, contributing to understanding how the organism was being transmitted and causing disease in healthcare workers. We released all of our protocols in not only the way we were detecting the virus in those early days, but also how we set the lab up. Contributing to the sequencing effort. So we linked up with Ian Goodfellow and others and allowed us then to take the samples we were taking from healthcare workers and plug them straight into the genomics facility. So therefore we were getting real-time sequences. We could take a swab from somebody, have a positive result within about 4 hours, and then have a genome sequence the next morning, which was a real kind of revelation at the time. And also then contributing samples to other things that we were doing in the way that our samples that we were collecting could contribute to other people's research as well. So it was terrible, but also quite an exciting period. And something then that I apologized to our collaborators for at the hospital was a kind of project that I didn't really want to do because I didn't think it would work. We've got this kind of fancy device which allows us to detect 96 different things in one sample. So we take a sample from a patient, we extract nucleic acid from it, and this allows us to detect any pathogen we want. So 96 different things in one go. What we did then is introduce these air filtration devices into the ward and had a kind of switch over design where they were off for a week, on for a week, and then off again. And what we could show is not only could we detect SARS-CoV-2 in the air of these spillover wards in Adam Brooks Hospital, but when we switched on these air filtration devices, not only did we obliterate the detection of SARS-CoV-2, but a whole host of different things that we didn't even realize were airborne also. So this is a real, again, interesting period of doing something that I didn't think would work, shows that actually having surveillance, real-time surveillance for what's circulating in airs in hospital has now become really important, and also shows that if you've got a friend that knows how to use social media, you can get it retweeted quite a lot and get quite a lot of publicity for it. Okay, so that's kind of what I didn't really want to do, but ended up doing, and it was an interesting period. So antimicrobial resistance, and probably we've heard quite a lot about this in previous years. Is it a big deal? Well, it kind of it's turned in the last 10 years of our understanding of what this means and the relevance of it. And there's been a lot of political power, which there hasn't been before, and it is a real problem. This is the O'Neill report from 2014, predicting that actually it's going to be probably the most devastating thing to hit humanity and with respect to disease by 2050, but also the associated economic cost. And we're well behind the curve on thinking about how we tackle this problem. So it's a problem that is growing. It's a real issue. As I said, working in Asia, it's a big problem in Asia, and therefore spilling over into other places. Okay, so this is then some examples. So as I said, there in the bottom there is Vietnam, where I was situated for 12 years. Most drug-resistant organisms spread from this part of the world. Why? Well, within that circle there, that encompasses about a 2.5 to 3 hour flight from Singapore, Bangkok, or where I was in Ho Chi Minh City. That encompasses about, I mean, it's mobile, but it encompasses approximately 1/3 of the world human population and about half of the world's animal diversity. So it's no surprise that new things, infectious diseases, drug-resistant organisms emerge from this location. And here's some work we've done at the bottom there, poor kind of animal kind of husbandry and poor kind of biosecurity on farms. They're highlighting a picture actually I took when I was there of the H5N1 outbreak. There's some plates up there. For those of you that aren't microbiologists, that is what a plate of a bacteria looks like. If you look at then the zone sizes around those discs, those are antibiotics pregnated into discs. If there's no zone around it, then it means that organism is resistant to that antibiotic. If there's a large area around it, it means it's susceptible to it. So this is then highlights the problem of AMR really quite beautifully. The reason being, this is a Staphylococcus aureus, and this is my Staphylococcus aureus that I cultured from myself and susceptible to antibiotic testing because I'd taken 3 courses of antibiotics and it was resistant to all of the antibiotics that I'd been taking for 2.5 weeks. Now, some of these, as you will know, as I've just identified, some of them are susceptible and some of them are resistant. So it is susceptible to some of these antibiotics. And I was given one of them, which then cleared the infection, but that was the last oral antibiotic I could have taken to cure that infection. I was about 2 days away from going to hospital and intravenous vancomycin for this particularly kind of benign skin infection. So it's not just that they become resistant to everything, it's access to certain antimicrobials that allow them to treat it quickly, that are rapidly available and cheap to use. And that's the problem. And this is something I'll come back to in a minute. That picture at the bottom right there, that is ciprofloxacin. Ciprofloxacin is a fluoroquinolone antimicrobial. That picture was taken from me walking around where I lived, so about 1.5 kilometer radius to about 30 different pharmacy shops and saying, can I buy ciprofloxacin? And all of them sold me ciprofloxacin. Okay, so why does AMR emerge and why it's a big problem? So antimicrobials and the way they interact with bacterial cells are really elegant and very sophisticated molecules with the way they inhibit or prevent something operating in the bacterial cell. But the way they work when we take them is not especially sophisticated. So they do not target individual organisms in us. So when we take an antibiotic or exposed to an antibiotic, any microorganism that we have on us or in us will induce some response against that chemical. Organisms that are susceptible to it will be killed by it, presumably, and organisms that are resistant to it will survive, but also they will induce some response to that drug. They have to interact with it in some way. And also when we take something, then actually the way it gets distributed through our body is not even, so therefore different organisms in our body respond to different concentrations of the chemicals in different ways, which makes it incredibly complicated. Okay, so as I alluded to, exciting working in Asia, and pretty much all drug-resistant bacteria, particularly the ones I'm interested in, come from Asia for the reasons I alluded to. So this would have been controversial some 10 years ago. Now it's not controversial, and actually hasten to remind you that we live in an interconnected planet where you can be from Delhi or Bangkok or Singapore or wherever in London within 12 hours, and these things get imported, and they get imported very, very quickly, and they move very, very rapidly. Okay, so let's hope this works. Oh, here we go. Okay, so this is then a time-lapse video, if you like, of a drug-resistant Shigella. Shigella is an organism that causes a very unpleasant bloody diarrhea, particularly in children. And then this then highlights the emergence of fluoroquinolone-resistant Shigella. On the bottom, we have a timeline where the blue dots are susceptible organisms, the red dots are resistant. On the right, we have a phylogenetic tree where we genome sequence the organisms and then we measure genetic distances between those organisms. And on the left there is probably more familiar, that's a map of the world. Okay, so let's have a look at what happens then. So this is work that we worked on for several years. This is published. If we then follow this, okay, we have then the emergence of a sampling broadly across the world looking at these organisms. We can see it populating the phylogenetic tree there. These are organisms that are susceptible to fluoroquinolones. Then approximately 2000 and whatever that is, 2010, we have the emergence of this resistant organism. So that's associated with 3 different mutations that occurred in one go. And then we see this radial expansion of it rapidly within the space of 3 years, radiating across the world and forming different subpopulations as it happens. And here, just to highlight, this is an outbreak associated with some patients in Ireland. This is how we picked it up in Ho Chi Minh City, but actually just to highlight my original points, not to point the finger of blame, but actually the vast majority of the organisms radiated out of South Asia, particularly out of India. So within 3 years, every Shigella that we isolated was resistant to fluoroquinolones, and it all occurred from one single event on the emergence of 3 individual mutations in one go. So what's the result? The result is pretty devastating, particularly across South and Southeast Asia. And you can read these articles if you so wish. Data there from an infectious disease hospital we work with in Nepal, showing the proportion of organisms that are multi-drug resistant. So that is resistant to 3 or more major classes of antimicrobials associated with bloodstream infections. Across the top there, different groups of different gram negatives and gram positives, all showing the same trajectory from about 2002 onwards. The middle plot is probably the most interesting. So the blue line maps the non-multi-drug resistant organisms. The red line maps the multi-drug resistant organisms. You can see there is a period where non-multi-drug resistant organisms are dominant. Then they kind of track each other. And then from 2010 onwards, then the multi-drug resistant organisms take over. I was in Kathmandu in May. If you were to look at this map now, then almost every isolate associated with a bloodstream infection in Kathmandu would be associated with some degree or other of multi-drug resistance. And this creates a problem. Okay, so a couple of examples. And I'll talk about the examples first and then go on to some of the ways that our laboratory is thinking about doing things about it. We're not the only people doing things about it, but obviously I'm talking, so I'm going to advertise some of the work that we're doing. It may not be the only way, but we'll talk about what we've done and how this may work. Okay, so some years ago, a journalist asked me what's the most dangerous organism that I've ever worked with. And actually, the one organism that I'm most concerned about is Klebsiella pneumoniae. Klebsiella pneumoniae is a gram-negative organism that lives in our gut. We excrete it on a daily basis, but it can cause really unpleasant infections, respiratory tract infections, bladder infections, but particularly bloodstream infections spreading to the liver and other places. Now, there are 2 different types that have been identified probably in the last decade. There's multi-drug resistant organisms. As I said, they're resistant to 3 or more classes of antimicrobials. And there's hypervirulent organisms. So these are bacteria that have the ability to spread and live in the bloodstream and cause these really unpleasant infections. So there on the left-hand side, hypervirulent associated with IUC locus, so negative or positive. And over the last 5 years, we've seen the emergence of organisms that are hypervirulent, but also resistant to ESBLs, These are extended spectrum beta-lactamases, so they're resistant to the most recent classes of cephalosporins, but also more concerningly carbapenems. So carbapenems are fairly serious injectable-only antimicrobials that are given only in the last resort. And we can see the emergence of those in the map at the top there on the right-hand side. You can see they're coming from India mainly. So these are hypervirulent organisms that are resistant to the last real choices of classes of antimicrobials that we have. And unlike the Shigella example that I gave, this occurs in multiple different ways. We can see MDR clones that acquire virulence plasmids, but also we can see these hypervirulent clones that acquire MDR plasmids. So this becomes a more fluid problem whereby the crosstalk within our gastrointestinal tract, we carry these resistance plasmids in different organisms, and then they can be transferred into a whole host of different pathogens. So The Shigella story, again, some works published a couple of years ago. Some complicated blots there. Those are plasmids. So plasmids are extra chromosomal bits of DNA that bacteria carry. So bacteria do have sex. They transfer bits of genetic information around them, particularly on these things called plasmids. And these are multi-drug resistance plasmids that have been acquired in Shigella. And what we found, which I think is really fascinating, if not terrifying, is that when you look in the general population from where these bacteria are circulating, we can identify identical multi-drug resistance plasmids circulating in the gastrointestinal tract of E. coli and other related organisms that are just hanging out, doing nothing, but they're carrying and circulating these genes. Now, what this diagram there on the plot on the right-hand side shows that if we go back and then give ciprofloxacin to these children, for example, or do this in a laboratory to mimic it, the action of ciprofloxacin stressing the organism means that we're more likely to move these plasmids from E. coli and other things that are just living in the gut into these pathogens. So by giving inappropriate antimicrobials, we can actually stimulate this effect of creating more drug resistance. By thinking we're doing good, we're actually doing more bad. So this then is an enormous, enormous problem, because we're not thinking just about the disease and how we treat with the disease, but we're thinking about the biomass of the microbiomes of human and animals and what's contained within them with respect to drug resistance. This is a cross-sectional study, relatively simple, looking at Vietnamese adults and children. And then this on the right-hand side, then each one of those represents a microbiome sample. So we take a stool sample from them, extract all of the nucleic acid from it, and then sequence it to look at the composition of the microbiome. All of this then corresponds to individual resistance genes to all hosts of different classes of antimicrobials that you or I would be given if we were seriously ill in hospital, meaning that actually all of those different classes of antimicrobial resistance genes are circulating on a pretty much everyday basis across most of Asia and probably a lot of us within this room as well. So the elephant in the room, if you like, to go back to that point, is that all of us then are reservoirs for creating and spreading drug resistance genes. Okay, so then a little bit more, talk about animals. This is the complex problem. They're all over the place. How do we deal with it if they're all over the place? Where are they coming from? How do we monitor this? What's the root of animals? What's the source of the environments? And then this call at the moment, which I don't necessarily agree with, a thought of a One Health approach, whereby we look at everything in one context, what's going on in animals, what's going on in humans, and also what's going on in the environment. And we've been involved in this particular project. This is in both Fiji and Indonesia, thinking about not medical interventions, but architectural interventions. So this is then a structure to then rebuild urban settlements in these places and try and think about the impact of how infectious diseases and also drug resistance may circulate in these places. And this is something we're getting to grips with at the moment. And we sampled then, oh, sorry, have I got data? I haven't got data. But what we've done then is sample a whole host of different environments in these places. So humans and animals, and also the households and the environment, to really get a grip on where the dominant locations are for the circulations of these pathogens and drug resistance genes in these locations. So it may be my slides, so I may come to that in a minute. Okay, so you may have seen the news at the weekend. Unfortunately, I got quoted. I wasn't misquoted, I did say this. So our health secretary has decided that from all the work that we've been doing to try and limit antimicrobials, that she thinks it may be a good idea to give them out in pharmacies, of which then I described as nuts and moronic, which then got in the Times. So this is, from what I've just shown you of this information, you will agree that this is a kind of moronic policy. The view of thinking about how we use these life-saving drugs and then letting pretty much have free access to them is crazy. So the use of antimicrobials, not use appropriate or misuse, it's the use of antimicrobials that impact on drug resistance. The more we use antimicrobials for anything, the more bacteria will become resistant. It's a natural evolutionary process. So the more we use them, the more likely we are to drive resistance. Oh, and this is the data from the other thing. So then this is the environmental information, again, showing understanding the quantity and the circulation of different resistance genes in these locations. Okay. So why is this important? And actually, this is a project we've been talking about this week with one of my colleagues. So just to go back about the thought of introducing or allowing the use of antimicrobials in the pharmacies, this is probably one of the best projects I've ever been involved in. It's not going to win me the Nobel Prize, but absolutely brilliant study of thinking about how empirical antimicrobial use occurs in the community. So actually doing a cross-sectional study, mapping the locations of pharmacies. So that is an area, District 8 in Ho Chi Minh City. It has a population of about 350,000, so about 3 times the size of Cambridge. And therefore, we can then map the population density to pharmacies. How close do people live to pharmacies, and what is their antimicrobial access? And also then by looking and doing this kind of mixed model, by doing secret shoppers, but also then by interviewing the members of the public about where they're likely to take their children when they have diarrhea, we can estimate how frequently children are being given antimicrobials for treating those infections in the community. And this is where we got the ciprofloxacin from. And actually what we found empirically is that actually they're more likely to go to hospital, and they're 10 times more likely to receive an antimicrobial in hospital than they are in the community as well. So then, therefore, this assumption that actually community access is a problem, it is, but also then hospital access is a problem because we don't have appropriate diagnostics. And if someone that ends up at hospital, they're really likely to receive an antibiotic on the basis of the doctor's feeling they have to treat them for something. So this, again, really interesting, and hopefully will it be expanded into other places of understanding how antimicrobial access works and what the impact is on AMR. Okay, so, I mean, slightly negative news, I guess, from where we are at the moment, but it's not all bad. I think that we've learned a lot, actually. I think the last 10 years have been a bit of a revelation. The dawn of bacterial genomics and other tools, I think we're really in a good position to think about not only how we understand this problem and estimate the magnitude of it, but also what we can do about it. And as I said, there are lots of different people tackling this problem as we move along. Getting pharma and other things involved is a bigger challenge, but actually we have lots of interesting leads into how we can kind of think about this problem. So what is it that we can do? So to me, as a microbiologist, studying this or thinking about this, then we could think about it as a kind of similar kind of magnitude problem as something like climate change. And what do we do about climate change? Well, at the moment, we're doing nothing. And there is an aspect of just sitting and waiting and seeing what's happened, as is kind of human nature, with the view of this may work in our favor, it may not. We're actually also really good at collecting information. So scientists collect information. We've been really good at kind of amassing information. There's a lot more information being collected, as I said, generation of genome sequencing data, generation of kind of intervention data from clinical trials. So we're getting better at generating data to really understand the magnitude of the problem, but it's not solving the problem. So what do we need to do? So drugs. We need new drugs. So we need to think about how we generate new approaches to kill drug-resistant bacteria. Diagnostics. So diagnostics are key. We can't really use antimicrobials appropriately unless we know what someone's infected with and how we treat it. Vaccines. So I think that if there is some good news from COVID, the introduction of new vaccine technology and the acceptability in some quarters of vaccines has become really exciting and has a lot of potential. Something I'm particularly interested, particularly with the Cambridge angle, is the use and development of therapeutic monoclonal antibodies, something we're getting into at the moment for understanding how we can target drug-resistant bacteria specifically with antibodies. Repurposing. So there's lots of different drugs out there that we use for different things, but we haven't really thought about how we can use them better or in different ways. Alternative therapies. A lot of people ask me, I'm sure someone will ask me at the end of this, as I always do when I give a talk about bacteriophage. Everybody's heard about bacteriophages, the viruses that kill bacteria. Can we use them? Will they be feasible? Something that's really scientifically challenging, but also very exciting, is we have a load of antibiotics and we know how to kill bacteria, but can we revert their activity? So can we suddenly come up with a technology that allows bacteria to become susceptible to antibiotics that we have? And something that is key, which again points out why I said it was moronic on the government suggestion that we should make them accessible, is to reduce the amount that we use and only use them when we really need them. But actually, what we really need to do is all of these things and not just one in isolation. This is not a one-stop shop. We need to come up with a whole host of different things and see how we can learn to tackle these major problems. And probably more. I think this is a really big area potentially for innovation, of thinking about how we tackle this particular problem from a kind of scientific point of view, but also from a social science point of view, from a clinical point of view. And actually, this is why I'm in Cambridge. So this is part of the reason I want to tackle this. I think there's lots of smart people here, and hopefully it's a challenge that I can, take on over the next 10 years of my scientific career. Okay, so I'm going to give you some examples of some kind of things that we've been doing, some things that we've learned, and then some ideas about maybe laterally about how we can think about tackling some of these maybe things indirectly. So my real kind of expertise, if you like, is in a disease called typhoid fever, which you've probably heard of. It's caused by the organism Salmonella typhi, and we've really kind of led the field in understanding the emergence and spread of drug-resistant Salmonella typhi over the last 15 years. Looking at, again, linking in with clinical trials. So rather than laboratory focus work, which we do, linking in with people being recruited into studies that are being given antibiotics, and then we actually study what happens to them. And over time, we can show in the middle there, increases to resistance to the fluoroquinolones, which then leads to the rejuvenation or generation of new antibiotics, or the use of new antibiotics to treat infection. Why is this important? Because typhoid has a 25% mortality rate without using antibiotics. So if you get one of these drug-resistant organisms to which you cannot treat, then you've got a 25% chance of dying. Over there on the left-hand side, again, opportunistic, but really interesting. During a clinical trial for a particular fluoroquinolone, we had patients that were in hospital for 3 weeks. We genome sequenced those organisms, and we found a new variant, a new mutation that arose during our study. And therefore, we could then predict how you were going to interact or recover or not from that particular drug just by sequencing your organism. So we detected these new mutations, which suggested it was resistant to this particular drug, and that correlated with how they would respond to the actual antibiotic. And then, perhaps more worryingly, the spread of those organisms over on the right there into sub-Saharan Africa. So this has also been a trope of other pathogens. Malaria is another one that spread. They emerge in Asia and then perhaps start attacking more vulnerable populations, particularly in East Africa. So we can show the emergence of these things, and then we can monitor how they move and how they spread. So then another kind of horror story, if you like, then is there's been the emergence of this organism called XDR typhoid in Pakistan. XDR stands for extended drug resistance. So this is resistant to even more antibiotics. So over there on the left-hand side, again, there's another phylogenetic tree looking at single nucleotide variants in the genome sequences of these organisms. And there's a big block in the middle that says Pakistan XDR. So this is, again, is an individual clone, a single organism that's spread that's resistant to all antibiotics, all oral antibiotics bar one. And that one is called azithromycin. Azithromycin is a macrolide. It attacks the ribosome. And if you look there on the left-hand side where it says Bangladesh ACRB and Pakistan ACRB, these are organisms that we've picked up that are resistant to azithromycin, so a single base pair mutation. So in Pakistan, we have XDR typhoid, but also we have typhoid that's emerging in a different location that's resistant to the last antibiotic we can give orally. And we found this in India, and this has been highlighted both by Gavi and the WHO, but also by Take On Typhoid, that we are essentially a single mutation away from having typhoid that is no longer treatable with oral antimicrobials. Okay, so what can we do? So what we can do then is just to highlight some of the things I was talking about. So a bit of a horror story and then some good news. We did some work with GSK looking at their compound libraries to try and identify novel antimicrobial agents. We screened 1.7 million compounds trying to identify things that may kill. We started with Shigella, as I alluded to earlier, but also then with typhoid. Out of those 1.7 million, we got down to 297 that actually showed any activity of killing the bacterium. Over there, that kind of plot shows that if you pick clinical isolates, so these are ones from patients rather than ones that have come from culture collections, you're way less likely to identify antimicrobials that do anything. So clinical isolates that are contemporary are way more resistant, even to drugs that have been locked away in freezers in GSK for 20 years. But we identified one. So actually, just to say, of these that we identified, not one was a single new mode of action. They were all known modes of action. They were just new drugs or drugs that had been locked away or used for different purposes. And we identified this drug called Tebupenin. Tebupenin is an oral carbapenem. And of those of you that are clinical, we can debate the use of oral carbapenems if you like. But what we find then is this has a really, really good activity to treat XDR typhoid and also XDR Shigella. And we're in the process then of doing some dose response studies in Nepal in typhoid patients to see whether we can actually use this oral carbapenem for emergency cases for people that are infected with these XDR variants, which is good news. But what is even better news is the introduction of a new typhoid vaccine. This has been around for some years, but it's only in the last few years that it's had enough momentum really to be tested. And I was lucky enough to be involved in these studies, a phase 3 trial in Nepal, but also in the final votes of the phase 3 trial, of which I can't remember now exactly, it's about I think 35,000 people randomized, and we could get 85% efficacy with this very low cost effective vaccine, which is a conjugate vaccine, which is the outside sugar on the bacteria linked to a protein to boost the immunogenicity. And the good thing about this is typhoid causes disease in children, and this can be used safely and effectively in children under the age of 2. And it's just been introduced in the last actually month or two. It's been put into the vaccination schedule routinely in Nepal. But also then, it was the justification for the XDR typhoid was a justification for the introduction of this vaccine into Pakistan. So there was an emergency case made, and then Pakistan adopted this vaccine to prevent typhoid on the emergence of these drug-resistant variants. And this has had a massive, massive impact on the circulation of both drug-resistant organisms and non-drug-resistant organisms. So if you prevent the disease, not only from the drug-resistant organisms, then we're therefore less likely to have the emergence of new drug-resistant organisms. So this has been fantastic. So it's been justified. So AMR has been the reason for vaccine introduction to Pakistan and Nepal, which wouldn't have done either. But what about other regions? How do we estimate disease burden? So something then we're thinking about is, okay, we've got this vaccine that works, but part of the problem of getting it used is understanding the burden of typhoid, because it's really difficult and complicated to set up studies to grow the organism from people sufficiently to demonstrate that it works. And what about other causes? So there are other organisms that cause a similar disease for which the vaccine doesn't work against, and are the different ways we can think about reformulating or changing the vaccine to do different things. And this is kind of where we are at the moment, and I'll show you a bit of data. Okay, so one of the key problems then with where we are with typhoid is not just having a vaccine, but it's how and where we introduce it. So is there a way that we can think about understanding how the organism interacts with the immune system that allow us to know whether someone's been infected with it, and that would allow us then to understand what the burden of disease by measuring it in a specific location? So this has taken probably 10 years of my life, because we don't really understand any of the outside structure of the organism substantially in the way it interacts with the immune system to identify these things. So we had to start from the beginning, both by looking at typhoid fever patients, but also people carrying it in their gallbladder like Typhoid Mary, to try and work out then what antibody response these people were making to different components of the bacterial surface. And we did this by a protein microarray, and we can show quite nicely that we can pick up specific proteins, and people generate an acute antibody response, but also then they generate a longitudinal antibody response as well. And how do we know what they do? Well, we can do various other things too. So we can see whether they generate what we call functional antibody. So these are antibodies that bind to the surface of the bacteria and actually trigger a downstream immune response. One thing we're particularly interested in, a response called a serum bactericidal activity. So the antibodies bind to the surface of the antibody. They bind something called complement, and the complement smashes a hole in the bacteria and kills it. So this is what we call functional antibody, which is really useful for a vaccine. And we've identified a whole host of different proteins that if we immunize mice with them, take the serum from mice, we can actually induce this serum bactericidal activity on the organisms, both for Salmonella typhi, which is the one we're interested in, but also for alternative forms that cause the same disease as well. So then suddenly we have the identification of antigens that are conserved across a whole host of different bacteria, that if we could formulate them into a vaccine, that it gives us a broader response, and we can immunize not only against the organism we're interested in, but also a whole host of other things. Okay, so let's go back then. Justification for vaccine introduction. A lot of work to then take serum samples from about 400 people that have had different forms of typhoid longitudinally over 3 months. And those graphs on the side there, some of them are flat. The ones we're interested in, the ones that kind of peak up, and the ones that kind of peak up and then drop down, this is a kind of conventional, what we call Sera Conversion. So you generate an antibody response to that particular individual protein, and that allows us to detect that that person has had a response to that infection. And we can combine a number of different components of the bacterial surface, so both proteins and polysaccharides, which behave differently. And these plots on the right-hand side show us that actually, if we combine them, it tells us different things about how we interact with the immune system, and that will tell us different periods of seroconversion against the bacteria. So we can combine them. So rather than just taking a protein and running our serum across it on a plate, we can combine them into an individual assay using this kind of Luminex technique, which allows us to put things on these tiny little beads, and they have a fluorescent tag on them. We can put them all in one go. We have all of these different antigens on these little beads, and then we can take these samples and then run them across anything we want. And just to demonstrate how important this is, we're just about to finish a study where we've run over 30,000 serum samples against these 10 different antigens. So it allows us to measure this at a scale which is absolutely enormous. So 300,000 data points. Okay, so then this is justification for thinking about how we use this again for vaccine introduction. This is a study from that was involved in across several different places. So this is information coming from out of Dhaka. This is what the freezer samples turned up. So these all turned up on a plane one day, and two of my research assistants had a heart attack when they had to unpack 30,000 samples from these locations. And then sampling these people that live in Dhaka, so a population of 100,000 people, we could then measure the incidence of typhoid in this location, which means that anytime anyone gets a fever in that place, they go to a hospital, they get a blood culture. If we grow the organism from their blood, they have typhoid, which is both cumbersome and cost-effective, and not cost-effective. And this is what it looks like. So we mentioned the incidence of typhoid over time from 2017 there to 2018. And we can see a peak there. So this is Salmonella typhi, and the alternative cause there is Salmonella paratyphi over the period of whatever that is, 18 months. So that's the proportion of blood samples that we get back that are positive, which is 0.2%, but also then the true incidence of measuring the proportion of people out of 100,000 that have typhoid over that period. And this is real data. Okay, this is people really having typhoid that we got the organism from the blood. And this is then what we get from our serology. So we take our different antigens and we run them. We take paired samples that are 3 months apart, and then we try and detect those that have an increase in antibody titer. So what we can see is ours map almost uncannily to the point of how those inflections occur in the true incidence. So the dotted line is the true data, and our colors are our different serological responses through time. So we can show that it maps really, really well. And remember, this isn't taking people's blood and measuring whether they've got an organism. This is just us taking serum from them and measuring an antibody response. And when we look at it by group, we can see here that how it declines with age, which matches the epidemiology. And we've got a number of different antigens there, which are probably telling us different things. But the key seroconversion events, or the biggest burden of disease potentially, is in children aged 0 to 4. Okay, so why is this then important? This then becomes really important for thinking about the justification for how we introduce vaccines. So we've shown then that drug resistance is a problem in typhoid. We can justify the introduction of the vaccine on the back of drug-resistant organisms, which has had a massive impact in Nepal, but we can't measure it accurately everywhere. This is now data for showing the true incidence. And if we look at the 0 to 4 age group there at the top, there's an adjusted incidence where we calculate the factor of how many people we've missed in the study. Therefore, we can estimate that just over 2,500 children per year in Dhaka have typhoid fever. Now, if we take our best antigen, which we think is this one, which is called hemolysin E, and we estimate then how many children have actually seroconverted over that period of time, our estimate then is 85,230 per year, which means that actually 85% of children are actually seroconverting to typhoid in Dhaka every year, meaning that the justification that the vaccine introduction is no longer whatever the crude incidence, 417, is actually 204 times greater. This is not disease. This is the amount of kids that are actually seeing that organism, drinking contaminated water, and then seroconverting. And then this now is the information that's then going to the WHO to justify the introduction of the vaccine in Bangladesh. So just some work on what we're doing and how this is working. We're working on this, we're trying to publish it, but also now it's been adopted on the fact we can use this, then we're trying to then release it internationally so people can then use it anywhere they want to. They can report this information back to WHO, and therefore we can justify the use of Gavi funds to use the vaccine in these places. Okay, so what else are the antigens used for? So There's a really clever vaccinology technique called conjugation. So lots of the things that we generate a vaccine for in bacteria are polysaccharides. They're sugars on the outside of the bacteria. They're very well conserved, but sugars are not very immunogenic and don't tend to generate very good protective immune responses. So what we do then is we attach them to things. We attach them to things called carrier proteins. And these carrier proteins are actually linked to the sugar. And when the immune system recognizes them, it processes them the same way. And therefore, you generate the same type of response to the sugar as you do to the protein. It's very clever. But these are a little bit complicated and expensive to manufacture. So working with a group in London and now someone in my laboratory, there is a mechanism for us to then bioengineer these ourselves using a clever little enzyme called PIGL-B and others, which we can make in bacteria. So what happens is we take these enzymes, we take our particular proteins and sugars that we're interested in on plasmids, we get them, put them in a bacterial cell, switch them on, and the bacteria themselves actually make conjugated vaccine, whereby they secrete protein and polysaccharide linked together into the periplasm, and we can isolate it, and therefore we can make very, very cheap vaccines because we get the E. coli to manufacture them. And we can take our proteins, which I alluded to before, which induce this serum bactericidal response, and suddenly not only then have we got the ability to make whole cross-reactive range of vaccines, but we can then theoretically link any protein to any polysaccharide. So if they want to come up with a new vaccine approach for any gram-negative bacteria, we potentially have the ability to pick the right protein and the right sugar, mix them, match them together, and then generate potentially an effective response against that individual organism or a whole host of different organisms. And just to show how this works, it's a little bit slow. So we've got them expressed. So we've got an E. coli bioconjugation strain with our particular plasmids in, and there you can see 1498. The others need some optimization. You can see that our sugar is in green and our anti-His protein is in red, and they link together. And we can show them we're actually getting the E. coli themselves to manufacture these vaccines for us, which becomes scalable in any form that we want. And again, what are we doing? We're trying to work on these. We're doing some immunizations with them at the moment, and we're doing some additional work to try and understand how variation of the different sugars and proteins may impact on this. Okay, and just one more thing then before I stop talking. Something that we're also kind of excited about is learning more about how we could use new technology to understand not only how bacteria become resistant, but also what we can do about it. We have this really exciting piece of equipment called an Opera Phoenix. This is a flatbed automated complex laser microscope, and we can run this in 96,384 or 1536 plates, which means that we can then put things into plates and analyze them at really, really high resolution. We can look at about 2,000 cells per well, and we collect about 80 images per bacterial cell, which allows us to do really detailed machine learning analysis on how bacteria are responding to drugs, also how they may respond to other interventions. And we're really kind of getting to grips with this now, and it's not become, initially it's become a kind of biological problem, but now it's become a mathematical problem of how we handle all this information and try and look at pulling out the important factors from the data we're generating. I just want to show you one example of what we're using it for. This is then an image. So as I said, we take a plane of these. This is a Klebsiella stained with different different stains, including cytox green, which allows us to tell whether it's live or dead. And then we merge them together, and we take 80 readings per individual bacterial cell of width and depth, of the location of the dyes, of all types of things, to try and work out how our different exposures are interacting with the bacterial cell. And this is then an example of just what we've done with this. So we can take this, we can take drug-resistant organisms, we can take drug-susceptible organisms, we can image them at high resolution at a certain time point, and we can separate them on the basis of certain changes in their morphology and work out what they are without ever exposing them to a drug. So we can identify changes on the bacterial cell surface that will predict how they're going to respond to a specific antibiotic without ever being exposed to the antibiotic. So the microscope can see things that you could never see with the human eye. We can measure these things in high level, and that allows us to infer how a bacteria will respond theoretically to any chemical thing we want to throw at it. And just then, thinking about how complex this is, to go back to the question of how we use this and how we generate new interventions. I showed an example of some of the vaccine work we're doing. Klebsiella is a really, really complicated organism to work with. It's almost impossible to find two the same. So this is then a phylogenetic tree showing a whole composition of different genes. It's really, really complicated. So how do we come up with a vaccine for a really complicated organism? And actually, we don't know, and we're trying to learn how different compositions, again, of sugars and proteins may interact with one another in one go to actually induce a protective response. So we have a broad collection of Klebsiella isolates, which lends itself to then us putting it through this high content imaging system. Our previous speaker mentioned outer membrane vesicles briefly on one of the slides. We take outer membrane vesicles, so these are natural components of the bacterial outer membrane. We immunize mice with them. We take them from different organisms, and then we can combine them together or look at how they generate an antibody response against the organism that they're from, or other organisms to see how broad or narrow that antibody response would be, which may induce a protective response. Okay, so then just a final example of how this works. We take our bacteria, we incubate it with our antibody or our serum from immunized animals. We image them in high resolution. We take our 80 readings per bacterial cell across these planes, and then we analyze the data. We can look then very, very simply at just looking at homotypic binding. So how the organism that you've immunized with generates a response against itself, but also heterotypic binding. How does it bind against other organisms? And then we can then really expand this then across a whole host of different collections to understand the different types of antibody we will get and how that then correlates with a particular response that we think is good for generating an appropriate immune response. And also then, okay, so how does this work if we were going to particular individual cocktail of outer membrane vesicles? We're then looking at these data in more detail. And this is data actually that we've just got within the last kind of week. This is then looking at all of those different organisms and the proportion of that bind those antibodies across the 6 different types. So there's 6 different serum here across our 75 different organisms, where we're measuring different variables, including antibody binding. And we can clearly see then that they don't all behave the same. And actually, if we were going to pick one, it would be a combination probably of number 4 and number 2, which show the broadest antibody response against a whole host of organisms that we could then combine to have a broader antibody response that may induce protection against a complicated collection of different isolates. Okay, so this is my final slide, and this is me to say thank you to my funders. Now, I do realize that's a bit of a whistle-stop tour of me showing perhaps some kind of tangential information about how we're thinking about AMR. So do we have the answers? The answer to this is no, we don't, but there's lots of different aspects of things that we're working on. And we will continue to collect data and describe data and use complex genetic information to understand how this works. But then by tackling this perhaps tangentially, I've showed you an example of how we're generating different vaccine approaches to understand broad and narrow antibody responses. I've shown you about a repurposing project trying to identify new chemicals or using existing chemicals, but also then using existing vaccines and actually generating the data on the back of antimicrobial resistance to introduce these vaccines on the basis of thinking about how we generate useful information can infer where they can be used. So these are my funders. So just to say, obviously my group at Cambridge, but also a whole host of different collaborators and places over the years. And these are my various funding organizations. And I'm contracted to say that I'm a Wellcome Trust Senior Fellow, and I've been delighted to be funded by the Wellcome Trust for the last 10 or so years. So any questions, I'll be delighted to answer them. Thank you very much. Is that on time?
Absolutely brilliant. Thank you. And it did get slightly more encouraging as the evening went by.
Yeah,
yes.
Don't take antibiotics unless you need them.
I think we got that.
Great.
Right. Great. I'm sure there are lots of questions.
It's quite a lot of information.
In the absence of antibiotics in a patient infected with a disease, are AMR organisms typically more dangerous than the pre-AMR? Are we making the organisms more dangerous in themselves rather than just not being able to rid a host of them?
We're not making them more dangerous per se with their virulence or ability to cause infection. We're making them more dangerous on the ability to clear them when they do cause an infection, and that's a problem. And it's happened because of the way we use antimicrobials as an insurance policy rather than a therapeutic in many occasions. Okay, so we generate this selective pressure on the bacteria that are on all of us. So then our conventional bacteria. So if you do this study, we have done, if you look at people coming into an ICU, and you probably do it in the UK, and look at what they get sick with, most of them get sick with their own organisms on the basis that we've generated resistance in them, or they contain resistance. They're immunosuppressed, they're given broad spectrum antibiotics, and they often succumb to infections of their own bacteria. Gentleman here.
Thank you. Do you think that vaccination is probably the more preferred route to eliminating these problems, rather than trying to find new ways of hitting the bacteria? And do you think it's particularly relevant to the east of the world rather than the Western world? I mean, does the success of vaccination in the East make life easier for us in the West?
Okay, so vaccination, yes, for some things. So typhoid's a great example. So it's a relatively, now we've got a vaccine that works. It's relatively straightforward to reduce disease burden, which is good. For other things which I alluded to, which are much more complicated, generating a vaccine against them would work in an effective way that would be competitive with antimicrobials. From where we are at the moment seems, I mean, it's a massive scientific challenge because they're so different. So how do you protect against a whole host of different organisms with a single vaccine construct? So unless we can think about how we generate broader responses to immunize against more, then for some things, no, probably. And with respect to how vaccines work, is it? Yeah, sure. So I think as a kind of social question, I think you're asking, yeah, the benefit of then thinking about this globally is really key. Because as I alluded to, most of these things come from other parts of the world. So if we can invest in understanding them and preventing them in other parts of the world, they're less likely to be an impact globally in the future. So yes is the answer to your second question. Come on, Philip, this better be good.
I think fascinating talk. I had one question that is more biologically related to antimicrobial resistance. So there are some theories that we have gotten antibiotic resistance before the emergence of synthetically produced antibiotics. And I presume because bacteria develop antibiotics to kill other bacteria, and other organisms develop antibiotics to, like fungi, to prevent themselves. Is some of these multi-drug resistant bacteria have emerged before the emergence of, let's say, human-produced antibiotics?
So there isn't an antibiotic that's been introduced where there hasn't been reported resistance within 3 years, which is a really rapid emergence of them. So, and it depends on the mechanism. So some of the ones that have been purified. So there's a difference between an antibiotic and antimicrobial. An antimicrobial is something that will kill bacteria that can be natural or chemically made. An antibiotic is something that's naturally produced that will kill bacteria. So antibiotics, such as the penicillins and other things, so these are derived chemicals from fungi and other things, as we know. So therefore, yeah, as a protection mechanism, some of these resistance things were circulating before, and we would then select from on the basis of the use of them, and that's happened. And as we then change those chemical classes somewhat, those enzymes evolve to keep up with the change in the drugs, and they get new mutations and new variants, and then we get them. So yes, that's true. Other things we've kind of, made and generated, the fluoroquinolones are a great example, and the macrolides. So these are chemically manufactured drugs that have been designed in the 60s. So they are not naturally occurring, and therefore those things have been generated pretty much de novo, either by mutations in the chromosome, whereby we select for them at low concentrations, or even enzymes that have emerged independently through the hybrid of different enzymes together, or different pathways that have kind of interacted with the drug in some way that then change and then can then interact and then either pump out the drug or become resistant to it. So the answer to that is both yes and no. So some things, yes. Some things we then actually have created new drugs ourselves and then created the problem ourselves afterwards. Yeah. So both.
Thank you. Apologies, I have two questions. Firstly, can we predict the mutations before they happen? And secondly, can we, sorry, do these organisms ever lose anything when they gain resistance?
Great question. Okay, so can we predict them? I mean, yeah, we can make them. I mean, it's an undergraduate experiment. We can take a low concentration of any antibiotic theoretically, expose it longitudinally to that particular drug in a laboratory, and generate some derivative that will have it become desensitized into it one way. So we know, if we know the target, then we can predict how it's going to change, and we can do that. Yeah, and there may be, again, scientific ways to protect that target a little bit to prevent mutation and stuff. That's one way. And the second question is, do they change alternative parts of the chromosome? Is that what you were asking? So do they have compensatory changes?
Yes, especially do they become weaker in any other way?
Yeah, so there's a big kind of thing about bacterial fitness. So, and the way I kind of like to think about it is, I used to tell my undergraduates when I was in Vietnam, if I turned up in Vietnam wearing my ski gear, okay, but I brought it all the way with me, it would be a negative. It'd have a fitness effect. I was carrying additional luggage for me that I wouldn't need, whereas if I went to Europe, I would. So they carry additional genetic information, if you like, that they don't necessarily need if they're not exposed to the drug. So yes, there is a fitness cost often associated with it. So therefore, some of them, these things are lost. So if we then stopped particular classes of antibiotics, they wouldn't disappear completely, but they diminish very, very rapidly, and that's been shown as well. The mutations are different. They are probably hardwired into the chromosome because they occur naturally at a certain rate, but it will probably take a long time for those to disappear. But drug resistance plasmids, depending on them and how they're being selected, which to be honest with you, we don't really understand either, some of it, then there is a fitness cost of those if you don't use a particular drug. Yeah.
Firstly, I want to say thank you for your lecture. And my question is, do we understand which mutations allow bacteria to become resistant to certain antimicrobials? And why do these mutations allow that change to happen? Okay,
so do we understand them? Yeah, we can identify them. So we know the binding location. So fluoroquinolones are a great example. So fluoroquinolones bind to the DNA gyrase, which repair kind of double-strand DNA breaks. Mutations in that particular codon 83 and codon 87, mutations in them then prevent the fluoroquinolone drugs effectively binding to the DNA gyrase protein. So we know that and we can predict it. Do they occur naturally? No, they don't occur naturally. So we don't find those even kind of spontaneous mutation rates without selection of the drug. So meaning that actually there is probably some negative effect of having those mutations if they're there. But because of the rate that bacteria mutate, without those, and the fitness problem of is probably so little that they probably become hardwired into the chromosome after a period of time, which we think is what happens.
Thank you.
Question from our side.
It's like an interview.
I don't have a question. I just thought you needed the exercise. I wonder if you could just comment on this. Last week, for the first time in my life, I was admitted to A&E. I went myself because I had a combination of symptoms which my GP was concerned about. I had various blood tests and x-rays and whatever in A&E, and they found nothing, except that the doctor gave me some antibiotics to take home and insisted that I take them. I said, can you just give me the prescription? If I'm not better tomorrow, I will go and get them. And she said, no, take them and take them all. I took them home and I haven't taken them and I got better within 24 hours. Are the medics doing this for fear of being sued, and do they need some legal protection to allow them to prescribe, if you like, at a reasonable level of risk?
I mean, I can answer that, but what about me being sued? So I mean, yes and no. So the problem is how we restrict them. So we've got so used to them being essentially as a prevention. So, you know, if people are going into an ICU that have a respiratory tract infection or whatever else, or a fever, then the assumption is they're likely to have a bacterial infection, which may be wrong. So, and often because of the way microorganisms interact with us, we don't necessarily have a diagnosis to identify what that is. If you go to Adam Brooks, for example, and they take a sample from whatever they think is you infected, even in somewhere like Adam Brooks, the chance of finding something is less than 50%, way less than 50%. If you do that in Southeast Asia, it's about 5%. If you get a diagnostic test, of which most places would never. So let's avoid the political ramifications of talking about what happens in the UK, so we can get over that. If you think about then what's going on in somewhere like India with a population of 1.4 billion people, where you can buy antibiotics over the counter for 10 cents. If you go to your doctor, then actually it's really cost effective for your doctor to take a different class of antibiotics, of the view that they're preventing or thinking they're treating you for an infection that's really almost impossible to diagnose. So this is why I alluded to having a really good diagnostic test, which is a massive challenge, because then you can pinpoint what someone's infected and know whether they have a diagnosis or not. I mean, there are methods to do this, but we're not very good at it. Just knowing whether someone's infected with a bacteria or a virus, for starters, of which, believe it or not, often we can't do. So therefore, the assumption is if you have a bacterial infection that progresses, that could be very serious for you. So therefore, it's easier to give a broad spectrum antibiotic. So yes, that does happen a lot. And that's one of the key problems. But I don't know your particular case, of course, I couldn't comment. Do they need more legal protection? Yeah, possibly. I think it's a brave doctor that restricts antibiotics on the basis if something serious happens to your patient. But then that study needs to be done. That trial needs to be done. And the downside of doing that study is probably you may have a fatality in one of the arms, which then becomes a problem with doing that study. What are the ethics surrounding that? And I think that's something we need to have a conversation about, probably about how we do those types of studies, not by giving new classes of antibiotics, but actually restricting them.
Can I ask a question about the economic case,
Yeah.
which is that it's hard, isn't it, with therapeutics, which are drugs of last resort, because often investment in it doesn't immediately see a return.
Yeah.
How do you see this as an inhibitor in your field of research and the application of research? And is there anything that you think that the venture capital industry or the drug industry or the biotechnology industry could do better to produce more effective therapeutics in your area of research?
I mean, that's an absolutely fundamental question. What an excellent question. Yeah. So the economics of, I mean, the economic argument for new antimicrobials is a disaster, because the cost of developing them, and there's been actually some really high profile failures with organizations, companies trying to invest in them and not gone anywhere. But then the current argument is that if you have a new antibiotic that will kill these XDR organisms that I've alluded to, you would only then give it to people infected with these particular bacteria, and therefore you wouldn't use it. The market comes from large quantities being sold to treat a whole range of different infections. So what do we do? Well, we probably need a complete rethink on the way the whole market is structured for understanding how we can actually then come up with something that's more attractive for pharmaceutical companies to enter into it, because that's clearly not going to work. So that doesn't work at the moment. And what can we do? Okay, so we have for example, I didn't talk about it this evening, but if I'm invited back, maybe I'll talk about it next time. We have an antibody which we think is really effective against a particular type of organism. Now, we could argue about the downstream costs of an antibody as a treatment, but if you think about then, as this gentleman said, going into a hospital and being given a broad spectrum antimicrobial, there is then quite a good economic argument if we have specific antibodies that target specific organisms that we know are circulating in hospitals, and therefore before you go in or as you're in hospital, you receive the antibody to protect you against that bacteria, which gives you instant immunity for the duration of your stay in hospital and beyond. And that becomes a different economic argument. And we have a lot of hospitals in the UK, we have a lot of hospitals in the US, where that would become economically viable on the basis of preventing against death or serious outcome by giving a prophylactic before people get sick, which then becomes a different argument, which probably is economically viable.
I think we've had a wonderful range of questions prompted by a spectacularly good, wonderful talk. I was going through waves of feeling pretty pessimistic, but actually the number of lines of things moving is very large, isn't it?
It is. There's a lot of exciting things going on, a lot of different technologies. I think, as the gentleman alluded to here, is when we find something, what becomes a pathway for turning it into something that becomes useful, and that's something we probably need to fix.
But there's sort of sense of whether we've sort of run out of runway or whether there are completely new approaches. The rate at which vaccines seem to become credible, we're much more optimistic about of late.
Let's see. I'll let you know in 20 years.
Okay, I was just trying to end on a really positive note.
No, I think I am optimistic. I think there's lots of interesting things. I'm not optimistic for antibiotics, but I'm optimistic for new technologies.
But you were giving that pitch that there are, we should do everything all at the same time.
Correct.
Which, by the way, is the right answer for climate change.
Yes.
Yeah.
So we need to do
Right.
something though, don't we?
Yeah, we do. But there isn't a silver bullet.
No.
Yeah. Right. Well, we can continue these conversations in the bar.
Sounds good to me.
Sounds a good idea. But Steve, thank you very much. It was
Thanks a
brilliant
lot.
talk.
Yeah, good. Thanks a lot.